Hello friends, today we are going to talk about oogenesis, the process of differentiation and formation of ovum from primordial germ cells. The major difference in sperm and egg is their size as well as their function. The process of spermatogenesis, we already talked about the process in our last lecture. When we compare their functions, we know that sperms are made up of nucleus and only a membranous structure and a specialized structure to transport the genetic material from male gonads to fertilization site. So they are simply the genome carrying machinery without specialized cytoplasmic contents. But ovum has very important functions besides the maternal genome donor. The ovum has to contain all the materials needed to initiate and maintain metabolism and further development. And that's why they have remarkably complex cytoplasm. When we talked about their numbers, the spermatozoa are produced in a very high quantity because of their very low probability of survival in external as well as internal harsh condition. We know that spermatozoa, they are foreign to the female body, and that's why the female also initiates or it starts a quick immune response against them in the form of leukocytes. But on the other hand, the eggs are produced in quite small number as compared to spermatozoa. Some species like sea urchin, frogs, they routinely produce hundreds or thousands of eggs because they have self-renewing stem cells. Self-renewing stem cells means a small portion is kept aside and they act as a stem cell, and remaining cells, they undergo the process of differentiation. Differentiation means formation of specialized cells from undifferentiated stem cells. Okay? Stem cells cannot perform any specialized function. That's why it needs to have a certain shape, and the process of differentiation gives it the particular structure and inter function, and that's why this process is very important. So when the stem cells are kept aside, that stem cells act as a reservoir, and that's why there is a continuous supply of stem cells, and due to that there is formation of that particular gametes over a very long period of time. While some species, just like humans and most of the mammals, they produce only few eggs as their oogonia divide to form a limited number of eggs. Now, when we talked about the germ cells and their numbers in humans, we know that there are the formation of this oogonia that start from 2 to 7 months itself. So during that time, the thousands of oogonia divide rapidly to produce roughly 7 millions of germ cells. After the 7th month of embryonic development, the number of germ cells drop suddenly, and most of oogonia die during this period. While the remaining oogonia means the oogonia which has not died, which survived here, uh, only those oogonia, remember their number is very small as compared to the 7 million here, and we can see the decline start here after nearly the 7 month. This is 6 month, this is 7 month. As of the 7 month of gestation, their number decreases, and the most of the oogonia die during this period, while the remaining oogonia enter the first meiotic division. These cells called as primary oocyte means the survived oogonia, we call them as primary oocytes. These primary oocytes progress through the first meiotic prophase. Remember, they undergo the meiosis 1, but it won't get complete. Instead, it arrested in meiosis 1, means in which phase? In prophase 1. Okay, and they are maintained in this stage until puberty, even until the onset of adolescence. Now, the group of oocytes that periodically resume the meiosis, so we can say that the first part of meiosis begin in embryo, and the signal to resume meiosis is not given until puberty, means they started the meiosis during the embryonic stage itself, but they have to wait for the signal to complete it. Okay, it is not like that the meiosis start and it get completed. No, meiosis start in the embryonic condition, it paused there, it halted there, and thereafter it resumed only during the puberty. So the primary oocytes continue to die even after birth of the millions of primary oocytes present at birth. Only 400 of these millions of oocytes, only 400 they are reach to the maturity during a woman's lifetime. Now this process, when we talk about the oogenesis, this entire process, this is the background. This oogenesis is divided into 3 phases. First, the germ cell migration. Germ cells, the initial cells that form gametes, we call them as germ cells, okay, or the gonadal cells. Second, the development of ovum, the actual process that is called as oogenesis, and thereafter the maturation of follicle. These are the 3 basic steps of oogenesis. Now we talked about the germ cells and their migration, the first part. So the primordial germ cells, the very initial cells, okay, that are going to be a germ cells. Okay, these are the primordial, means the cells which are going to form germ cells. Yes, they make their entry on here yolk sac. Okay, on the 24th day of fertilization, these primordial germ cells multiply by mitosis, and once they reach the gonadal ridge, okay, now we can see these primordial germ cells here. They are moving here along with this forming hindgut, and they reach the region of a genital ridge. Remember, these primordial germ cells, they are still in their undifferentiated states, and it means that it has potential to form either ovary or testis. This stage is called as bipotential stage of gonadal development. So what determines whether it is going to form a testis or ovary? And it is the testis determining gene or testis determining region of Y chromosome, that is especially the SRY gene and the male specific gene such as SOX9. If they are present, then it is going to form testis, otherwise it is going to automatically leads to the femaleness. So depending on its chromosomal composition, it is going to form, this bipotential gonad is going to form either testis or ovary. Now during this embryonic life, these primordial germ cells, they are also known as oogonia, they interact with somatic cells. Somatic cells, obviously the somatic cells, they are present here. Remember, the somatic cells which are original cells that are present in this genital ridge, and these primordial germ cells, they are the migratory cells. They have migrated from the yolk sac to this. Okay, so these primordial germ cells, they interact with these somatic cells, and mitotic divisions of primordial germ cells it starts. But remember, it is without the incomplete cytokinesis, means no complete cellular division, and this leads to the formation of germ cell nest. It means that this incomplete cytokinesis, okay, and it's just like a syncytia occur there, and these primordial germ cells that surrounds this nest. So while this process is going on, some oogonia in this germ cell nest undergo death, okay, and that creates a small space there, and that allow the somatic cells to surround this individual oogonia, okay? And due to the surrounding of these somatic cells, there is formation of a follicular cells, and this is how the primordial follicle forms. Remember, these primordial follicles are still diploid in nature, and in order to make them efficient gonads, it has to undergo the reduction division, that is meiosis. Okay, and this meiosis began in embryonic period itself, remember, and it has arrested in prophase 1 and remember arrested until puberty. We already talked about it. Periodically, a group of primordial follicle enter a stage of follicular growth, and during this time the oocyte undergo approximately 500 fold increase in their volume. You can see here that there is formation of this primordial follicle, and during their development it has increased in its size, and also there is an increase in the number of these surrounding cells. We call them as the granular cell, and which form a concentric ring around the oocyte. Throughout this growth period, the oocyte remain in the dictate stage, means the prophase 1. The fully grown follicle thus contain a large oocyte surrounded by several layers of granular cells. We can see here the granular cells, they are in the concentric layers, and the innermost layer that stays with the egg here. The innermost layer is there that form the cumulus or corona radiata, which surround the egg during the growth in the oviduct. So during the growth of follicle, even a cavity occurs there, that is more clear in this. So you can see here a small cavity start appearing there and it increases. This cavity is called as antrum. The antrum is for holding antral fluid, is a mixture of proteins, hormones and other molecules. So at a given time during this periodic cycle, a small group of follicles get mature. So this is how the follicles are formed. So during the menstrual cycle, means at a particular time during a periodic cycle, a small group of follicles get mature. Okay, however, not all of them survive. For surviving, these follicles must expose to gonadotropic hormones at a correct stage of their follicular development. So as these primary follicles capture the gonadotropin hormones, it divides its nucleus. We call it as the germinal vesicle. It breaks down and the metaphoric chromosomes migrate to the periphery of cells. So the actual process of this oogenesis start here. So at that telophase, one of these two daughter cells contain a very small amount of cytoplasm, whereas the other has nearly the entire volume. So we can see here when this primary oocyte it get exposed to the gonadotropin, it resume the meiosis and it divides. Okay, so you can see they are not having the equal volume of cytoplasm. One is having quite bigger and another one is having very small volume of cytoplasm. So the first one is here the secondary oocyte and another one is called as polar body. During the second meiotic division, again there is an unequal cytokinesis occurs, and the second polar body uh, is formed here. Okay, so the secondary oocyte divide into a mature ovum and one small cell, we call it as secondary polar body, and the first polar body it also divide here and it form two polar body. So in total a primary oocyte forms one mature ovum and three polar bodies. Remember, the meiosis 2 means here when it resume. Again, let's have a revision. The primary oocyte, it start at meiosis 1, it completed during the puberty, means during a menstrual cycle when the gonadotropins are there. So that is the meiosis 1. But meiosis 2 resume only after fertilization process complete, remember. And now this is a timeline. You can hear the things are more clear here. Actually, this is the revision. So these are the primary germ cells. Okay. Remember this 2N that indicates their chromosomal content. This form the oogonia. They start their meiotic entry. The meiosis start here. Okay. And it gets suspended here. That is a meiotic prophase 1 in this dictate stage or dictate arrest. Okay, and it remain suspended during the puberty. Then this primary follicle start their development. We call it as a folliculogenesis. Okay, when it start when they get exposed to the FSH, follicle stimulating hormone, that is at the time of puberty. Then this follicle develops, it forms antral cavity, and then there occurs ovulation. Still it has not completed meiosis 2, it is still in meiosis 1. So after ovulation it completes its meiosis 1 here, and then after the fertilization process occurs, we can see here the sperm is there. When the sperms make its entry into this egg, the fertilized egg completes its meiosis 2. This is also the revision here. The primary germ cells are there. Then primary germ cells move to the bipotential gonads. Then depending on the sex, I mean depending on the chromosomal content and the genes, then there is this differentiation, and then this meiosis occurs. Then this meiosis is suspended there. Then the follicles are there. These follicles undergo the ovulation. Ovulation means the meiosis 1. Meiosis 1 completes only after the FSH surge is there, and it completes its meiosis 2 only after the fertilization. So now let's have a talk about the hormonal control. So in first part of the menstrual cycle, we called it as a proliferative or follicular phase. The pituitary secrete FSH in quite higher amount, follicle stimulating hormones. So any maturating follicle that reach a certain stage of development respond to this hormone. Remember, not all follicles. Okay, follicles we already talked about that these follicles they are growing here unless and until they reach at a appropriate stage when they are able to respond to this FSH, then and then only there occurs the growth of these follicles. Now this FSH also induce the formation of LH receptor on granulosa cells. So these are the granulosa cells. When this follicle respond to the FSH, it start the expression of LH receptors also. The LH, the luteinizing hormone, it's going to be a very important function in the oogenesis, especially during this release of ovum from the follicle. So shortly after the initial period of follicular growth, the pituitary begins secreting LH, and this LH surge broke the dictate stage, meiotic stage, and it start or it completes meiosis 1. So again, have a look here. We can see these are the follicles here, these are the growing follicles here, and when these follicles grows, when they respond to this FSH, this green line is the follicle stimulating hormone. Meanwhile, the luteinizing hormones, its concentration is less, but this increase in the FSH causes expression of LH receptors here, and after some time this LH increase here. Obviously, means already their receptors are there, and now these follicular cells, they respond to this LH hormone, and that causes ovulation. So now let's have a talk about the hormonal control. So in first part of the menstrual cycle, we called it as a proliferative or follicular phase. The pituitary secretes FSH in quite higher amount, follicle stimulating hormones. So any maturating follicle that reach a certain stage of development respond to this hormone. Remember, not all follicles, okay? Follicles, we already talked about that these follicles, they are growing here unless and until they reach at a appropriate stage when they are able to respond to this FSH, then and then only there occurs the growth of these follicles. Now this FSH also induces the formation of LH receptor on granulosa cells. So these are the granulosa cells. When this follicle respond to the FSH, it start the expression of LH receptors also. The LH, the luteinizing hormone, it's going to be a very important function in the oogenesis, especially during this release of ovum from the follicle. So shortly after the initial period of follicular growth, the pituitary begins secreting LH, and this LH surge broke the dictate stage, meiotic stage, and it start or it completes meiosis 1. So again, have a look here. We can see these are the follicles here, these are the growing follicles here, and when these follicles grows, when they respond to this FSH, this green line is the follicle stimulating hormone. Meanwhile, the luteinizing hormones, its concentration is less, but this increase in the FSH causes expression of LH receptors here, and after some time this LH increase here. Obviously, means already their receptors are there, and now these follicular cells, they respond to this LH hormone, and that causes ovulation.